Today in What's New in 22Q, we're taking a deep dive into some really groundbreaking research.
Yeah, it's quite exciting stuff.
Stuff that brings, well, a lot of hope, I think, and a fresh understanding of 22q11.2 deletion syndrome. Our goal with these deep dives, as always, is to give you a shortcut. Cut through the complex science language and get straight to the important insights. We want to help you grasp those aha moments that could really make a difference. And today, we're zeroing in on a specific study, a really pivotal one that looks into a potential early intervention strategy for 22q. We're focusing on this fascinating piece of work. It's titled Oxytocin Seals the Blood-Brain Barrier, Improving 22q11.2 Deletion Syndrome Trajectories. The first author is Giulia Castellani, along with her colleagues, and it was published recently in 2025 in the journal Brain, a top journal.
Absolutely. And our mission today really is to unpack all of this research. Make it clear, make it helpful, translate these, complex findings into insights that actually matter for you and your family. We don't just want to know what they found. We need to understand why it's so significant for people with 22q. And, importantly, what new possibilities this might open up down the road.
Okay, let's get into it then. Let's unpack this. So many of you listening are very familiar with 22q11.2 deletion syndrome, or 22q for short. And you know the range of challenges it can sometimes bring. Things like social and intellectual difficulties, maybe a higher risk for conditions like ADHD or schizophrenia later on. But what's really illuminating here, what this research digs into, is something maybe less discussed. It's how the genetic factors in 22q interact with the body's immune system, and crucially with the brain's own protective barriers. It's a really interesting new lens.
It really is. And to investigate this intricate connection, the researchers used a specific tool. They used a specialized mouse model called LGDIL plus mice. Now, these mice are genetically engineered, basically, to have a deletion that very closely mimics the human 22q11.2 deletion.
Right, so it lets them study things you can't easily study in humans.
Exactly. It gives a controlled environment to look at how the syndrome progresses and, importantly, test potential interventions. And what they saw in these mice was, well, pretty compelling. They found that adolescence really seemed to be a critical period. That's when certain behavioral and brain changes really started to kind of emerge and become noticeable.
Okay, what sort of changes are we talking about?
Well, we saw difficulties with social interactions, for instance, things that look quite similar to challenges sometimes seen in humans with 22q. They also showed issues with something called sensory motor gating.
Sensory motor gating. Can you simplify that a bit?
Yeah, basically it's how the brain filters incoming sensory information, like sounds, sights, touch. It helps you tune out the irrelevant stuff so you're not overwhelmed and can focus.
Okay. So like a filter to prevent overload.
Precisely. And these mice had deficits there. They also saw, starting in adolescence, this gradual decline in social abilities, and alongside that, disruptions in how key brain areas were developing, especially an area called the prelimbic cortex.
And that's important for...
Oh, it's vital for social behavior, for decision-making, emotional regulation. So changes there, they could potentially explain some of the behavioral patterns.
That makes sense. So you've got this picture in the mouse model, these challenges emerging in adolescence. What did the researchers do next? Where did they look for a possible intervention?
Well, this is where it gets really interesting. They turned to oxytocin.
Oxytocin. Okay, many people have heard of that. The love hormone, right?
That's the popular term, yeah.
Yeah.
And it definitely plays a big role in bonding, trust, social stuff.
But it does more.
Oh, much more. Its functions go way beyond that. It influences brain development, how we handle stress, even regulates the immune system. It's a remarkably versatile molecule.
Okay, so they decided to try giving oxytocin to these 22q mice. How did they do it, and when?
So they administered a small amount of oxytocin intranasally.
Through the nose?
Yep, basically through the nose. And here's the really striking part. They did this very, very early, just for 1 week, from day 1 of life, the day they were born, up to day 7. That's it.
Just 1 week.
Right after birth.
Just 1 week. An incredibly early and, importantly, non-invasive treatment.
Wow. And what happened? Did it work?
It had genuinely long-lasting beneficial effects. It was quite remarkable. This short, early treatment significantly improved their social behaviors later on. They interacted more typically. It also helped with that sensory motor gating problem we talked about. Their brains got better at filtering information.
That alone sounds significant.
It is. But there's more. It actually helped normalize the development of brain structures. Remember that prelimbic cortex we mentioned? Yeah, the one important for social behavior and decision-making.
Right. This early oxytocin seemed to prevent some of the thinning and disorganization that they saw in the untreated 22q mice in that area, it essentially set their development on a more typical trajectory.
That's really powerful.
Yeah.
Just 1 week of treatment having effects that last into adolescence.
Exactly. It really highlights this idea of a critical window of opportunity very early in neurodevelopment.
So the big question then is how? How does oxytocin actually do this? How does it protect the brain? You mentioned the study points to the blood-brain barrier.
That's the key mechanism they identified. Yes, the blood-brain barrier.
Okay, can you break that down for us? What is it, simply?
Absolutely. So imagine your brain is like a fortress, a really important, sensitive place. It needs top-notch security. That security system involves these barriers. The main one is the blood-brain barrier, the BBB. There's also a related one, the blood cerebrospinal fluid barrier, or BCSFB.
Okay, 2 barriers, like layers of security.
Kind of. Think of them as highly selective filters, or maybe very strict bouncers at the door of the brain club. They meticulously control what gets in from the bloodstream and what stays out. Their job is to protect the brain from harmful stuff. Toxins, pathogens, even rogue immune cells floating around in the blood.
Okay, so very important protection.
Huh.
And what's the issue in 22q, according to this study?
Well, the study suggests that in individuals with 22q, and they saw this clearly in the mice, these protective barriers can be a bit leaky.
Leaky.
Yeah, more permeable than they should be. Things can get through that shouldn't. And this seems to happen because there are lower levels of certain crucial proteins. They're called tight junction proteins.
Tight junctions, like the glue holding the barrier cells together.
That's a great analogy, yes. Think of the cells making up the barrier as bricks in a wall. These proteins, like one called Claudin-5 in the BBB and another called Claudin-1 in the BCSFB, they act like the mortar between the bricks. They seal the gaps, making the wall solid. If you don't have enough mortar, or the mortar is weak, the wall gets leaky.
Okay, that makes sense. So the barriers in 22q might be leaky because there's less of this mortar.
That's what the evidence points to.
And oxytocin, how does it fit in?
This is the really cool part. The study found that oxytocin directly helps to seal these leaky barriers.
Like applying new mortar.
Pretty much.
Yeah.
It seems to boost the levels of these critical tight junction proteins like Claudin-5 and Claudin-1. So it strengthens the barrier, makes the filters more robust, more effective.
And when the barriers are sealed properly.
Well, then fewer unwanted things can sneak into the brain from the blood. Specifically, the study mentioned certain immune cells called myeloid cells.
Okay.
If these myeloid cells get into the brain when they shouldn't, they can cause trouble, contribute to neuroinflammation.
Ah, so sealing the barrier stops these cells getting in.
Exactly. It prevents that infiltration. And by doing that, the oxytocin treatment also helped reduce inflammation in the brain, particularly in areas like the medial prefrontal cortex, which is, again, crucial for complex thinking and social stuff. So sealing the barrier improved the whole immune environment within the brain. It stopped that buildup of pro-inflammatory signals they saw in the untreated mice.
Wow, so it's not just one effect, it's like a chain reaction starting with sealing the barrier.
It appears so, a really fundamental effect.
And it's just remarkable, I have to say again, that this treatment was so early, just the first week of life, and so brief. Yet the protective effects lasted right through development in these mice, into adolescence, even adulthood.
It really underscores the potential power of intervening very early, targeting these fundamental protective systems.
Okay, so let's shift gears a bit. What does this all mean for you listening at home? For families, for caregivers of individuals with 22q? What are the practical takeaways here? Or maybe the future possibilities that this research opens up?
Well, first off, the discovery itself is really significant. Knowing that 22q might involve lower natural oxytocin levels in the brain, and that the blood-brain barrier is more permeable, that's a huge piece of the puzzle.
It helps explain things.
It helps us understand some of the underlying biology. Yes, it could contribute to why there's sometimes increased brain inflammation or issues with social and cognitive development in 22q. And the fact that oxytocin seems to directly address this leakiness well, that's incredibly promising. It points to a new mechanism, something fundamental happening very early on that might influence the whole developmental path.
So promising. But this was in mice, right? How hopeful should we be about this applying to humans?
That's always the crucial question, of course. This was a mouse study. But the findings are really encouraging. And importantly, they're very consistent with what we already know about the biology and challenges in humans with 22q11.2 deletion syndrome. So while we need human studies, this strongly suggests a potential new therapeutic avenue using oxytocin. It moves us beyond just managing symptoms, potentially.
So what could that look like in the future? You mentioned new avenues for treatment.
Well, this research really opens the door for future clinical trials in humans. For instance, you could imagine studies where oxytocin is given to babies diagnosed with 22q, given very early on, maybe even shortly after birth, like in the mouse study.
To do what? To replicate the mouse results.
The hope would be to achieve a kind of neuroprotection, to use oxytocin to help seal those brain barriers right from the start, supporting healthier brain development from day one.
Neuroprotection. That sounds huge.
It could be. Imagine the possibility of a non-invasive early treatment, something that helps create a more secure, less inflamed brain environment right from the beginning. The potential impact could be preventing some developmental challenges before they even fully emerge, or at least significantly reducing their severity.
So potentially smoother cognitive development, better social skills.
Better overall well-being throughout life, potentially. That would be the ultimate goal.
What are the steps to get there? It sounds exciting, but also like there's work to do.
Oh, absolutely. Rigorous clinical trials in humans are essential. We'd need to confirm these findings, figure out the right dosing, timing, and most importantly, ensure it's safe for infants.
Of course, safety first.
Always. But this mouse research provides a really strong foundation, a strong rationale to actually start planning and conducting those trials. It gives us a clear target, the brain barriers, and a potential tool, oxytocin, to move towards an intervention. An intervention that could maybe enhance the brain's natural plasticity and help maintain healthy function long-term for individuals with 22q.
It really is an exciting prospect. This idea of using something non-invasive, like intranasal oxytocin, so early, it makes you wonder, could oxytocin act as a general sealing factor for brain barriers in other conditions too?
That's a fascinating thought. Many neurodevelopmental disorders are suspected to involve issues with brain barrier function or neuroinflammation.
So this could have broader implications.
Potentially, yes. It definitely shifts the focus, doesn't it? From just managing symptoms later on to possibly addressing a root cause very early in life.
That feels like a paradigm shift. It's definitely a hopeful step forward.
A very hopeful step, I agree.
And we should mention, this deep dive has been brought to you by 22q11 Europe. They're the European association supporting families living with 22q11 deletion syndrome, and they're dedicated to bringing you the latest, most relevant research like this. So wrapping up, this study really offers a fresh perspective, doesn't it?
It certainly does.
On how we might support individuals with 22q. It emphasizes that early intervention window and really highlights this complex link between genes, the immune system, and these crucial brain barriers. It leaves us with a powerful thought, I think, that targeting these fundamental protective systems right at the beginning could potentially unlock significant improvements in development for people with 22q. Definitely something for all of us, especially you listening, to think about how these early insights might shape the future of care.
Thank you for listening to What's New in 22q, produced by 22q11 Europe. Take care and talk to you soon.